Good morning to everybody. So today we are going to discuss about metabolism of drugs. So first we will see that introduction. Second, what are the phases involved in the metabolism? So, two phases involved in the metabolism of drugs. Phase 1 and phase 2. So these phase 1 and phase 2 is mediated by some enzyme. They are called cytochrome P450 family. So we will discuss one by one. What is biotransformation or drug metabolism? Chemical alteration of drug in the body that converts non-polar or lipid soluble compounds to polar or lipid insoluble compounds. In another way, we can say the chemical changes undergone by drugs within the body are referred to as drug biotransformation. The majority of these chemical alterations results from the interaction of drug with some enzymatic system. They are called endogenous enzymatic system and non-enzymatic system. First we will see what is the consequence of biotransformation. Here the active drug will be converted into inactive metabolites. The example will be phenobarbital and morphine. Secondly, due to that biotransformation, the active drug converted to an active metabolite, phenacetin. Example, phenacetin. And then third, due to biotransformation, an inactive drug, it will be converted to an active metabolites, levodopa. So now we will see what is prodrug. When an inactive precursor is metabolized to a pharmacologically active drug, the parent compound is known as prodrug. So this prodrug name was coined by Albert in 1958. What are the advantage? So this prodrug will be used for increased absorption of drug, elimination of some unpleasant taste, decreased toxicity, decreased metabolic inactivation, increased chemical stability, and prolonged or shortened action. So before going for phases of metabolism, we can see where this biotransformation or metabolism takes place. The liver is the major organ responsible for biotransformation of drugs and other xenobiotics. Kidney and other organs may sometimes be involved in biotransformation. The product formed by biotransformation are mostly excreted by kidney, less frequently excreted via feces and expired air. So here the metabolism of drugs takes place in two phases. One is phase 1, another one is called phase 2. Phase 1 and phase 2 biotransformation or metabolism of drug reactions are mediated by enzymes located in various subcellular components such as cytoplasm, mitochondria, lysosomes, and microsomes. Here, Phase 1 reaction takes place in three events. 1. Oxidation. Number 2. Reduction. Number 3. Hydrolysis. Phase 2 reaction, these are the conjugation reacting involving compounds such as glucuronic acid, glycine and glutathione. So what are the compounds eliminated from that phase 1? That compound will be conjugated with in phase 2 reaction. That conjugated molecules as glucuronic acid, sulfate, acetate, glycine and glutathione. These are compounds which are involved in the phase 2 reactions. So here that phases of metabolisms takes place in two events. One is called phase 1, another one is called phase 2. That phase 1 metabolism, it include hydrolytic reactions, oxidation and reductions. In phase 2, that conjugation takes place in the phase 2 reactions. So in phase 1 reaction, here that P450 monooxygenase system will be involved. So that is called cytochrome P450 enzymes. These enzymes are heme protein having a single ion coordinated to protoporphyrin called as heme. So this heme undergoes reduction, oxidation during the catalytic cycle of enzymes. So site of drug metabolism. Here oxidation and conjugations takes place by extrahepatic microsomal enzymes. They are located in the brain, lungs, liver, intestines and some other organs also. Another enzymes called hepatic microsomal enzymes, they also involved in oxidation and conjugation reactions. It will be takes place in the lungs. And then another enzymes called hepatic non-microsomal enzymes. It is very important. They are involved in the liver. That means phase 2 reactions will be mediated by these hepatic non-microsomal enzymes. Example, glutathione, glycine and glucuronic acids. So phase 1 are non-synthetic reactions. So phase 1, it will be mediated by the three reactions: oxidation, reduction, hydrolysis. Conjugation will be taken by phase 2 reactions. In phase 1, that oxidation takes place. So here that oxidation is nothing but addition of oxygen or negatively charged radical or removal of hydrogen or positively charged radical. So these reactions are carried out by group of enzymes called monooxygenase. This monooxygenase enzyme is located in the liver. So in the liver, that phase 1 reactions of oxidations takes place. And then what are the components involved in that phase 1 of oxidation reactions? Cytochrome P4 heme protein. So it act as the prosthetic groups, NADPH, cytochrome P450 reductase enzyme and molecular oxygen. So what is that monooxygenase? So this is a major enzymes which will mediate that oxidation reaction in phase 1 stage. Here it will be catalyze a drug and oxidation takes place in the liver, kidney, GI tract, skin and lungs. Oxidative reactions occur cytochrome heme proteins, reductase, NADPH, phosphatidylcholine and molecular oxygen. So where actually these cytochrome P450 enzymes are located? So they are located in the smooth endoplasmic reticulum. So here lot of P450 families enzymes are present. So some of the enzymes which will be metabolize that drug. So here that cytochrome P450 and other enzymes of that drug metabolism microsomal systems, they are encoded by super genes called mammalian nuclear DNA. So here that P4 system families include 74 CYP gene families. Totally they are involved 74 CYP gene families. They are named as CYP 1, 2 and 3 involved in drug metabolism in human liver. Especially that 1A1 we can name it as that CYP1 A1. It will be metabolized that theophylline and caffeine. Second CYP1A2. So these P4 system enzyme metabolize caffeine, acetaminophen and the phenacetin. CYP2A6. So these metabolize testosterone, and then CYP2B6 it metabolizes cyclophosphamide, erythromycin and testosterone. And then CYP2C families. So these cytochrome P450 enzymes metabolize lot of drugs. Example there is given in tolbutamides. And then CYP2E1, so they metabolize caffeine. CYP2C9, so it will be metabolized brufen and tolbutamide. And then another important enzyme CYP2E1. So these CYP2E1 metabolize alcohol. So these are the some enzymes, P450 enzymes, which are involved in metabolism of some drugs. Next we will see that non-cytochrome P450 drug oxidations. So here that two enzymes which are involved in non-cytochrome P drug oxidations. One is monoamine oxidase, another one is diamine oxidase. This monoamine oxidase located in the mitochondria, and it will be deaminates endogenous substrates including neurotransmitters. Example dopamine, serotonin, norepinephrine and epinephrine. It is also metabolized alcohol and then alcohol dehydrogenase. Non-specific enzymes found in soluble fraction in liver, so they metabolize that ethanol. Some of that flavin monooxygenase, they require molecular oxygen, NADPH, flavin adenosine dinucleotide, which are involved in the drug metabolism. Next will be called that reduction reaction. It will be takes place in the phase 1. Here reduction reaction is nothing but conversion of oxidation. So drugs primarily reduced here that holothionein or chlorohydrate. Third stage is called hydrolysis. It is also takes place in phase 1 reaction. Here The cleavage of drug molecule taking by molecule of water, that is called hydrolysis of water. Where it will be takes place? It will be takes place in liver, intestines, plasma and other tissues. Example, cholinesterase, ionized pethidine, oxydoxine. So the event oxidation, reduction, hydrolysis, all these three things will be takes place in phase 1 reaction. After, the drugs will be eliminated due to these three reactions. It will be enter into the phase 2 reactions. In phase 2 reaction, that eliminated compound will be conjugated with the compound which are present in the phase 2 reactions called glutathione, glycine and glucuronic acid. So here you will see that phase 2 synthetic reactions. Conjugation of drug in phase 1 metabolite which an endogenous substrate to form a polar highly ionized organic acid. In phase 2 reactions, the events takes place in number 1, glucuronate conjugation, methylation, acetylation, sulfate conjugation, glycine conjugation, glutathione conjugations. Here in phase 2 reactions, conjugation with the glucuronide, conjugation with the glycine, conjugation with the glutathione. So these events will be takes place in phase 2 reactions. Glucuronide conjugations. So quantitatively, the most important phase 2 pathway of drug and endogenous compounds. Products are often excreted in the bile. The enzyme which will be involved in here, glucuronylysyl transferase. The compounds with a hydroxyl or carboxylic acid group are easily conjugated with glucuronic acid, which is derived from glucose. Enterohepatic recycling may occur due to gut glucuronidase. So these enzymes excreted in bile can be hydrolyzed by bacteria in gut and reabsorbed and undergone some fate. This recycling of drug prolong its actions. Example, phenolphthalein, aspirin and morphine. Amino acid conjugations. Here that amino acids involved in conjugations, glycine, glutamine, arginine. So here glutathione conjugation. This glutathione is a protective factor for removal of potentially toxic compounds. Conjugated compounds can subsequently be attacked by gamma glutamyl transpeptidase and a peptidase to yield the cysteine conjugate. The product can be further acylated to N-acetylcysteine conjugate. So that example is paracetamol. What is that Hoffman eliminations? Finally, inactivation of the drug in the body fluids by spontaneous molecular rearrangement without any agency of any enzymes. So this is called Hoffman eliminations. So here, when oral dose is considerably higher than sublingual or parenteral dose, when marked individual variation in the oral dose due to differences in the extent of first phase metabolism, the oral availability is apparently increased in patients with severe liver disease. The oral availability of drug is increased if another drug competing with it. Chlorpromazines example. Thank you.